with that, thank you for coming, and they're all yours.
Great. Well, thank you for inviting me, and I look forward to talking with you. And what I'm going to do today is really give two separate presentations. The first one is going to talk generally about the Cancer Center and how we support cancer research, particularly translational cancer research within the Cancer Center. And then I'm going to talk about a research project that we've been working on in my lab for about 25 years at one level or another, that really speaks to both the challenges and the promise of translational research. And then I'm going to leave about 15, 20 minutes at the end for discussion. So we'll have time to talk and answer questions. My disclosure is that I do research that's supported by Checkmate Pharmaceuticals, and I'll be talking about some of the research that's related to that in the second half of my presentation. So here are the learning points. The first is to understand how the Cancer Center supports the full spectrum of basic clinical and population-based cancer research, and the translational research that links those different types of research. And then second, I'll dig into the science and talk about a cancer immunotherapy that's undergone quite a few twists and turns that I've been working on, as I mentioned. So the goal of the Holden Comprehensive Cancer Center is really threefold. We support research, we support clinical care, we support education. And that includes everything that we think might impact on the burden of cancer. So it starts in the research laboratory, it goes to the patient, all the way to the community. And so we have researchers looking at basic genetic processes that make cells grow at the most basic level. We have a large number of clinical trials, I'll talk to you about that are ongoing, many of which are available only at our Cancer Center. And then we work in the community as well, and this is a heat map looking at advanced colorectal cancer, with red areas being a high burden and blue areas being a low burden. And this is equally important type of cancer research. If we can figure out why some areas and some populations are suffering a disparate burden of cancer, we can sometimes go in to address that. In this case, we identified the fact that here in the southeast corner of the state, the primary care physicians were not screening for colorectal cancer the way they should have been. And we were able to intervene, and that's a different type of translational research, but still incredibly important. And then, as I said, we're involved in research, clinical care, and education, all the way from prevention, early detection, therapy, and quality of life. And then my job as director is to try to tie all this together and to get people from different fields, totally different backgrounds, but with a common interest, looking to reduce the burden of cancer. So that's the goal. We all know of the three-legged stool that we talk about in academics, where in order to do good research, you have to have bright students around, you have to have patients that are willing to participate in the research, and equal in the other areas, we really can't do any of these things well unless we do all three of them well. So if you look at how we support collaboration, we really do it in multiple different dimensions. And I've been told by my colleagues I shouldn't have a solved Rubik's Cube here. I should have one with lots of different colors on all the different sides. We start out with our basic research programs. And these are the programs we present to the National Cancer Institute as how we organize our research. So we have a program called Cancer Genes and Pathways. That's our basic science program. Two programs that are designed to be translational, experimental therapeutics, and free radical metabolism and imaging. And then our population science program. And each of these contains investigators from multiple colleges. As you might guess, the College of Public Health, including Bill, is located in the Cancer Epi and Population Science program here. But we also organize ourselves around cancer types. And we came up with a terrible name, and I take responsible for this, of the MOGs. And the MOG is a multidisciplinary oncology group. And this is basically a group of clinicians and researchers interested in a specific cancer type. So my research is mostly in lymphoma, in the experimental therapeutics group. So if you look at where I would fall, it would be both within the experimental therapeutics group and in the lymphoma MOG. And the MOGs not only provide clinical care, multidisciplinary clinical care, but they come up with the ideas for clinical research by seeing patients. They know what unanswered questions there are and what kind of trials we need to do. And they also work with basic and population scientists who have an interest in that particular cancer type. Then finally, we have some cross-cutting teams that don't really fit into either of those categories. We have a team that does bone marrow transplant and cellular therapy. You might have heard about CAR T-cell therapy, a very exciting new form of immunotherapy. We have a team that does that. We have a team that focuses on the psychosocial well-being of our patients, on palliative supportive care, et cetera. And so each of these different ways of organizing are different ways of supporting collaboration and translation. So we sort of think of this as a diamond, with everything starting with the basic research. And a lot of the work going on in cancer genes and pathways isn't specifically designed towards a therapy or towards a prevention. But we know that by doing basic research, we come up with discoveries that can have an impact. And the example I'm going to tell you about is one of those examples. We then have our 2 translational programs that have both basic scientists and clinical investigators in them. And this is where we take concepts that come out of the basic science, we develop them, get an idea of how they might be useful clinically, and move them towards that application. And then finally, where the rubber meets the road is when we take these discoveries and apply them to where they can really help people. And that's where the cancer epidemiology and population science program really comes in. And then we have collaborations that work across this full network. And in fact, we have had retreats of between 2 programs with each one of the 2x2 combinations you can have here. And you can think about how collaborators would work across those disciplines to do things together that they can't do by themselves. We have a lot of researchers that are involved in the Cancer Center. These are mostly faculty members who are doing research that's cancer relevant. And you can see there are a large number of them. And they come from 6 different colleges and 36 different departments across the university. Not only that, we collaborate with researchers from across the country. One of the organizations we helped found is called the Big Ten Cancer Research Consortium. We realized that as we learn more about cancer and the molecular nature of cancer, if we're going to target a specific genetic abnormality, for example, that might only be present in 2% of people with a given cancer. You're never going to be able to study that at one cancer center. By working together, we can open trials and get them done more quickly and get answers for patients. So the Cancer Research Consortium now has every one of the Big Ten cancer centers as part of it. We have 21 open trials that are opened up across the network, and we're able to do translational research faster that way than by working alone. And we've also worked to establish collaborations across institutions. So in some cases, we have a basic scientist at Purdue working with a clinical investigator at Iowa. Neither of them could do it alone. By working together through the consortium, we encourage that kind of collaboration. These programs, as I already mentioned, work across the MOGs so that we have people who do free radical metabolism and imaging, but work on a particular cancer type here. And you can really cross vertically and horizontally and find a researcher in every one of those positions that you can look at. And then we rely very heavily on shared research resources. And in fact, our large grant from the NCI that supports the Cancer Center, more than half of that funding goes to support these cores. And these cores provide expertise to the researchers across the institution. And the 2 that have the biggest footprint in the College of Public Health would be the Biostatistics Core and then the Population Science Research Core led by Brad McDowell that I don't think I have on this slide. But those are based in the College of Public Health, yet are used by people from across the Cancer Center. And the same is true of the other cores. So when it comes to clinical cancer care, one of the unique characteristics of our Cancer Center is that we really provide multidisciplinary care. That patients will come in, and more often than not, they'll actually see different specialists in the same place on the same day. And that allows us to really bring together all these experts to have input into how best to manage the patient, who is always at the center. And then we have 14 different tumor boards where clinicians from different specialties and pathologists and radiologists will meet, talk about a case, and really discuss what's the best management for this patient. Should they get a new novel immunotherapy on a clinical trial? Do they need surgery? Is this an early stage cancer that doesn't need therapy that can be monitored? And by getting all these experts together talking about that, we can come up with the best opinion for the patient so they don't have to go to 3 different doctors and get 3 different opinions. And this is also, as I said, where the idea for clinical trials come up. Because it's very common for a patient to be presented at a tumor board and for everyone to agree that we don't know what the right answer is for that patient. How should we manage this person based on the research that we know about that's going on? And if we don't know, very often that's the nidus for developing a new clinical trial and a new concept. And then these groups also have basic scientists who participate, who can actually contribute and say, well, wait a minute, this patient might have this genetic abnormality, or there might be this cause associated with it, and to help that move forward. And then we have one of these for each of the different types of cancer. So we have experts who do nothing but study lung cancer and take care of lung cancer patients. Same with breast cancer, et cetera. And this kind of subspecialization allows our faculty to really understand what's going in our disease, but they also collaborate with other people so that ideas from one field can be taken to another. We have a clinical trials support corps that now has over 60 full-time staff that work to help support clinical research trials. And if you look at our accrual to interventional clinical trials, that continues to grow. We're expecting to hit over 600 patients this year that are enrolled in these types of trials. This takes an incredible amount of work with respect to IRB approval, making sure they're scientifically rigorous, making sure that we're following things are being done ethically and that the patients understand the trial and are consenting to participate, making sure we're tracking the data effectively, making sure that's being monitored. All of these things take a huge amount of effort to do this right. And yet it's the way we move things forward. When we sum up all of the cancer research that's taking place at the institution that's supported by peer-reviewed funding, that comes out to about $25 million a year of funding to our members. That doesn't all come through the Cancer Center. Some of that comes through departments, but we facilitate it and we help support it. And then we also have collaborations with industry and other groups, so the total approach is $34 million. So it is an incredibly exciting time in cancer research right now. In fact, a lot of the basic research that's been done over the past 3 decades is finally paying off. I started in the field of cancer immunotherapy 30 years ago, and any number of the world's experts in cancer told me I was throwing my career away because there was no way to get the immune system to fight cancer, and that it was a waste of my time. But in fact, a lot of people working very hard, mostly in the basic science labs, have finally helped us figure out how we can use the basic science of immunology and immunotherapy to help patients. And that's not the only area where we've seen progress, but in fact, that investment in translational research, starting with areas that you really didn't quite know how it would help, is really where it all starts. We are able to treat patients, and in some cases cure patients now, that we couldn't have dreamed of just 10 years ago. 10 years ago, a patient with metastatic melanoma, that was a death sentence. Now we can cure, not all of them, but a significant percentage of those patients with immunotherapy. And other cancer types are starting to show the same story. And this, again, is due to basic research in the laboratory, getting that into patients, and now we're working to make sure that patients in the community getting cancer immunotherapy are getting it right. A point I always make is that the best of times and worst of times, one of the challenges has been that as people have gotten excited about cancer immunology and immunotherapy, more researchers are putting grants in. And so what's happened is the pay line at the NCI has plummeted. The chance of any one grant getting funded is now in the range of 10 to 12%. So ideas that are great sometimes won't get past the pay line and get funded. And so this is one of the challenges we work on. And in fact, a lot of our work is supported by philanthropy from grateful patients and other supporters of our effort. And another point I make is that our speed of progress really will be proportional to investment. We have a lot of great ideas that are out there, and we've just got to figure out how to support them so that they can help patients. So now I'm going to change track completely and start talking about research and really highlighting the long and winding road of research. And this is from eastern Iowa. That's not working. And I liked it because it's a winding road, but actually it's not very accurate because I don't see a lot of turnoffs and I don't see a lot of alternative places to turn left and right. So this is probably more accurate. This is what translational research looks like. In fact, this is what a lot of research looks like, where you're faced with a lot of options and you make your choice and you try a lot of different things and they don't work and they don't work, but you keep going and then finally you get to where you want to go. So let me tell you a story that sort of is like that. Now this is the linear version. So this whole story started with a colleague here who now has started a company based on this. All right, I'll just use the arrow, named Art Krieg. And Art was a basic scientist, a rheumatologist actually, who was trying to develop antisense DNA to turn off B lymphocytes. Okay. And what he found in a control well is that the B lymphocytes were growing like crazy. So he thought, oh, it's contaminated. The company blew it when they made the oligo for me. He threw it out. He made another one. It did the same thing. He went to a different company. It did the same thing. So he started playing with the sequence of that piece of DNA. And he found that, in fact, that piece of DNA was immunostimulatory and activated the B cells and found out it activated other cells as well. So an unexpected finding. This wasn't research geared toward developing a therapy, but it really got him thinking, what could he do with this? And I happened to have a lab right around the corner. And since I was a cancer researcher, I said, well, let's see what this does with cancer. And so went ahead and did some preclinical studies to look at what effect these short sequences of DNA had on the immune system. And that led to clinical trials, and that led to additional formulations that are now being evaluated in the clinic and showing promising results. And I'll go through this a little bit more. This is the linear version. This isn't how it happened. This actually started much earlier. Once Art found this, he went back to the literature and found the case of a surgeon in New York named William Coley. And this was back in the late 1800s. And Dr. Coley had heard the case of a 7-year-old girl who had a large tumor in her neck and got infected with what they call erysipelas. Whether it really was streptococcal infection, we don't know. Back in those days, it was more based on symptoms. And her tumor shrank away. And he talked to some of his colleagues, had they ever seen this? And they said, yes, they've seen this now and then. So what he did is something you couldn't do today, but he took live bacteria and injected it directly into tumors of some of his patients. No human subjects review, no informed consent. He just went ahead and did this. And some of the patients died from the infection, but some of them, the cancer regressed. And he then went ahead and decided, well, maybe if we kill the bacteria, it won't cause infection. We'll have the same effect. And he saw it did in a few patients, but not many. And other people tried to repeat it. As you can guess, different bacteria, different patient types, different ways of injecting it. And so this was always out there in the literature that this could work, but it was sort of thought to be pseudoscience for about a century, with no understanding of the mechanism of how it might work. So that brings us to Art's serendipitous discovery. So what Art had found is an oligonucleotide, a piece of DNA that has sequences that are very similar to bacterial DNA. And I won't go into the details, but it has to do with both the base sequence and whether or not they're methylated. And they're called CpG-ODN. And the CpG-ODN make the immune system think that there's a bacterial infection. The immune system gets activated in the absence of an infection. And that can then result in subsequent effects. And a few years after Art's discovery, it was learned that the receptor for the CpG-ODN is called toll-like receptor 9, or TLR9. And furthermore, not all pieces of DNA are alike. There are different classes of the CpG-ODN. So this all sounds like very basic work. What is the translational implications of this? Well, the translational implications are that if you can stimulate the immune system, maybe you can actually get the immune system to fight cancer. So we actually went ahead and tried to see, could you immunize against the cancer using this CpG DNA as an immune adjuvant? And this is a paper we published back in 1997. The answer was yes. We did this in mice. Could we use it to enhance how well monoclonal antibodies worked in the mice? And the answer was yes. When we inoculated a mouse with a tumor and we treated them with the monoclonal antibody plus the CpG DNA, more mice survived than those that we treated with just the antibody alone. And then we identified the cells that were responsible for this. That it's a type of cell called a dendritic cell, which was just being described at that point in time. And furthermore, when you activate those dendritic cells, they can activate T cells that can attack the tumor. So all of this sounded really exciting and said, okay, now we've got an idea on how we can use this basic science discovery and translate it to the clinic. So again, this was about 5 years worth of work here. So we did that. And we went ahead and first did a phase 1 clinical trial. And these are patients for whom no other therapies were available at the time. And we found we could give these agents safely to patients. We gave them IV. And we saw some evidence of immune changes in the patients. And maybe we saw a little slowing of their lymphoma, but nothing to write home about as far as it being a therapy. But we showed we could give it safely. We showed it had immune effects. We then combined it with anti-cancer antibody in patients who were otherwise going to get the anti-cancer antibody alone. And again, we found it had immunologic effects. We found maybe a hint of a therapeutic effect. But when we crunched the numbers, it would have taken a study of about 1,200 patients based on the statistics to confirm that it enhanced the therapeutic effect of the antibody. We couldn't get anybody to agree to that trial. So we had the biology. We thought we had something going on, but we were unable to take it to the next level. And we even tried to combine it with radioimmunotherapy, with antibodies to which radiation is attached, in this case, yttrium-90, to try to show that the combination was better than either therapy alone. And again, it's the same story over and over again. Hints that it worked, but no home run, nothing to write home about. So if I look at this 15 years of studies that we worked on, and I summarize them on a single slide here related to this translational research, we found that these agents have both in vitro and in vivo effects in both humans and mice on malignant B cells and cells of the immune system known as antigen-presenting cells. That they're very dependent on the sequence of that DNA, and we had dissected that out and looked at hundreds of different sequences to try to figure out which sequence was best. We also realized that how you delivered it, how often you gave it, what the regimen was, what the cancer type, were all important questions. And this actually speaks to one of the major challenges in translational research. If you have an idea, where do you go with it first? Because if it doesn't work the first time, sometimes you don't get a second chance. And so you really have to think carefully with limited data and limited knowledge, how can I learn as much as possible, as quickly as possible, to encourage movement of this idea so it might actually help people someday. We did find out that these agents have a very short half-life, that they last for 1 to 2 hours. And that was a real challenge, that we could give them, but we have nucleases in our blood that are trained to destroy bacterial DNA. And so we give these agents, and we can stabilize them by tweaking the backbone, but not too much. So that was another problem. And then in each one of these studies, we knew we were doing something, but we weren't quite there yet. So this led us to step back and think, well, what are we doing wrong? How can we rethink what we're doing here? And actually, part of that was going back to Coley and saying, okay, what did he do that's different than what we're doing? One of the things he did differently is he injected his bacteria directly into the cancer. We were giving it systemically to try to stimulate the systemic immune system. So that led us to explore this idea of what's now called in situ immunization. We know a lot more than we did in Coley's day. Back then, no one knew anything about antigens and T cells and immune responses. But what we know now is that one of the challenges in cancer medicine is that every patient's cancer has a distinct repertoire of antigens. Many of these are neoantigens that are only expressed in the cancer and not in other tissues. But those neoantigens are different in every patient. So you can't go to the shelf and take a flu vaccine off the shelf and make a cancer vaccine and give it to a patient, because the protein there is going to be different in every single patient. So how can we deal with this? Well, one way is to try to turn the tumor itself into a vaccine, change the tumor microenvironment in a way that the immune system itself can recognize what neoantigens are in the tumor. What is it different about that cancer that would allow the immune system to come in and fight? And so that's known as breaking immune tolerance. So the immune system that was ignoring the tumor now says, wait a minute, this tumor has a neoantigen that doesn't belong. We're going to fight it off. And so by injecting these toll-like receptor 9s into the tumor, we can change the tumor microenvironment, hopefully in a way that allows the immune system to come in and fight it off. Now, we also knew we had a problem that even when we did this, the half-life of these pieces of DNA was very short. So we, again, took a step that was sort of a quarter of a step back towards what Coley had done. And instead of using the naked piece of DNA, we made a virus-like particle that contained this DNA. And this actually is composed of a bacteriophage capsid. So this is a protein that naturally self-assembles around any nucleic acid. So if we take the capsid and we throw it in with our CpG DNA, we get these beautiful virus-like particles that have no infectious capability at all, but are real potent stimulators of the immune system. And the bacteriophage capsid naturally protects that DNA because as it does with the bacteriophage DNA. So it doesn't get degraded until the particle actually disintegrates. So Art Krieg, who I mentioned came up with this discovery, at this point actually had developed a company, and we worked with him to develop these virus-like particles. So we decided to give them the shot. And just to recap, we know they're taken up by antigen-presenting cells. We know that their release of this immunostimulatory agent takes place over a longer period of time instead of being very, very quick. And by giving this foreign protein itself, that's an extra danger signal to the immune system. And we found in a series of studies over a couple years that these virus-like particles work better than the naked DNA. I will skip that and go directly to some of the mouse studies. So these are studies where we inoculated a mouse, in this case on one side, and then we injected the tumor either with just saline or with our virus-like particle. And this is a very aggressive lymphoma. With the virus-like particle, we were able to cure about 20% of the mice. And the goal is not to cure the mice, but to show that it has a therapeutic effect. Even more exciting is that if we took a mouse and we injected it on both flanks with tumor, so it had two lymphomas growing, we only injected one side with our therapeutic agent, we saw that the tumor on the contralateral side began to shrink. So we were doing something systemically that was having an anti-tumor effect. And when we went ahead and depleted the T lymphocytes from these mice, the therapy didn't work. So if you look here, that without the T lymphocytes, the therapy is totally ineffective. You have to have both the CD4 lymphocytes and the CD8 lymphocytes. And then we also found that it works synergistically with another form of immunotherapy that's been getting a lot of attention lately called anti-PD-1 therapy. So this was all we thought pretty exciting, that we're finally, now, of course, we had found in the mice that the other forms of TLR9 therapy had worked pretty well too. So we knew that we had to move on. When we looked at the tumors, we found that the therapy enhanced the immune system inside the tumor. This is staining for T lymphocytes. So we got an infiltration into the tumor of the T lymphocytes. And I'll skip that. So this is when the translation really, really took place, when we moved into the clinic. And this study was led by Dr. Millam, who's our leader of our oncology division and head of experimental therapeutics. And it was a multi-center study, but we were the lead center. And it was supported by the Checkmate Pharmaceuticals, that is Art Krieg's company. And we studied whether or not this approach could work in patients with advanced melanoma. And these are patients who had been through all sorts of therapy before, really had no other options, and in fact had already been treated with immunotherapy called anti-PD-1 and were resistant to it. So this was a group of patients really who were in tough shape. The inclusion criteria included metastatic and unresectable disease. They previously had not responded to anti-PD-1. It was a dose expansion study, so we started at a lower dose, moved up, and then once we hit a dose we thought was good, that was the dose we stuck with, so it was a phase 1-2 study. So I'm going to show you 3 different kinds of data here. We call them a waterfall plot, a swimmer's plot, and a spider plot of the patients here. So this is the waterfall plot, and this shows all of the patients what happened to their tumors. And this is a group of patients who, again, really didn't have any other therapeutic options. So without therapy, you'd expect the vast majority of them to have tumors that grow. But as you can see here, a significant percent of them had tumors that shrank. And shrank pretty considerably here. Now this is a best response. In order to qualify as being called a partial response or complete response, you need to maintain that response for a certain period of time. So per these criteria, 22% of patients had a response, but in fact, it looks like more than that percentage actually benefited. Another question was, did we see shrinkage of just the node that we injected, the tumor mass that we injected, or like in the mice, did we see a distant cancer start progressing? And in fact, we saw both. What we saw, these are all the patients who responded, that the injected node, the mass is in green, the non-injected is in light blue here, and you can see we saw shrinkage of both. And in some patients, they just had a single very large tumor, but some had local and distant tumors, and they both shrank. And then perhaps most gratifying of all is what we call a spider plot, which showed us how long did these responses last for in these patients? How long did they benefit? And what you can see here is that among those patients who had tumor shrinkage, many of those tumors continued to shrink over time. And the number of patients who had shrinkage and then regrowth is quite limited. And this is what's different about this kind of therapy compared to some other forms of cancer therapy, where you can induce resistance by giving the treatment. The immune system, once it starts fighting off the cancer, tends to be plastic and can identify additional neoantigens. And some of these patients now are in continued remission 3-4 years after starting on this trial. So we're incredibly excited about the potential here. This is an example of one patient who had a lot of metastatic disease all over her body, mostly in the skin. And this was the lesion that got injected with the agent. You can see she had regression of her other tumors. If you look really carefully, you can see it's pale around these lesions. That's called vitiligo, which happens sometimes when the immune system attacks pigmented cells. So it looks like we now not only were getting this immune system to attack the melanoma, but it was also recognizing some antigens in those normal pigmented cells, particularly near where the cancers were. And so that's a particularly interesting case. After a few months, we did a biopsy on one of the lesions because it still had a little bit of pigment in it. And what we found is the cancer was gone, but some of the macrophages had a melanin still in them. And so this patient remains in remission now a couple years out. And when we looked at the expression of T cells, when we look at T cells in the tumors, we didn't get too many biopsies, but we found that some of the tumors indeed had T cells had infiltrated into the tumor. I mentioned combining it with therapy. One of the things we did do is we induced upregulation of a molecule called PD-L1, which was the rationale for combining it with antibody that blocks the effect of the PD-L1, and I won't go into all the details there. So we're now taking this to the next step. This study is closing shortly, and we're following the patients and writing it up for publication. We have a lymphoma trial that should be opening any day. We've gone back and forth with the IRB over some trivial things, and we're ready to open it up in lymphoma. There is a trial going on in lung cancer. We're now looking in melanoma at neoadjuvant therapy. So if we have a patient with a large melanoma who's just been diagnosed, who we know if we do surgery, their chance of relapse is 60-70%. So we can cut it out, but the chance of it coming back is very high. What if we inject that tumor with our agent before we surgically remove it? and induce the immune response, so the immune response can kill off the micro-metastatic disease that might be there. So this trial is ongoing. We're seeing 75% of people have the local tumor disappear with the therapy. We don't know yet whether it's going to reduce the incidence of long-term relapse. That'll take longer for us to figure out. So this is another example of translational research where you start out in the most challenging population because ethically that's the way to do it. You take what you learn then and see if you can help more and more different groups of patients. And we're looking at other cancer types as well. And we're continuing to look in the lab at some of the other questions that will allow us to make this therapy more valuable. And that's called reverse translation. So what you do is you take an agent that you've developed in the lab into the clinic, you see what it does in patients, but then there are some unanswered questions in the patients that you can take back to the lab and do translational research in the lab, trying to make that therapy even better. So it's a continuous circle that goes around. One of the things we're doing now is trying to understand using modern technology, such as single-cell RNA sequencing, what effect are we having on different immune system cells that might be in the tumor microenvironment by giving this therapy. And we've already found some surprises, but we're still trying to figure out how to analyze this data. So this is just one example of the winding road of translational research. Starts out with an exciting idea and anecdotal evidence that it can work. And in our case, that was over a century old. That's usually followed by attempts to duplicate it that result in frustration and failure. That's due to technological limitations, lack of understanding of basic science, and the fact that we just don't know enough to understand why something works or doesn't work. And we're there with a lot of the therapies we have at this point. It's not uncommon for enthusiasm to wane, and that's enthusiasm in the scientific community, enthusiasm in the public sector, enthusiasm in the private sector. You couldn't get a company to invest in cancer immunotherapy 20 years ago in any way, shape, or form, and now everybody's on board and the money is pouring in. And colleagues in the field, as they did with me, told me I was chasing a failed hypothesis and was throwing my career away. And then something happens, and you start to be able to figure it out, and you're able to move forward. I suspect we're going to recycle, that we're going to end up with frustration and failure as we move forward, and we're going to have to work our way through it. So here we are. We've got Coley, 100 years. Came up, identified CpG DNA as being something that looked like it was interesting. We did preclinical studies showing it had anti-tumor effects. That got us excited, and we went to early phase clinical trials. Those didn't turn out so well, so we went back to the lab to figure out if there was a better way to use these agents. This went on and on and on and on, with the hope that eventually you'll have clinical applicability and be able to help patients. Whether we're there or not, I don't know, but I am really excited about it. So the folks who have done all the work within my lab are listed here, along with the sources of support. And then the clinical trial that was led by Mo included investigators from across the country who collaborated on all these studies as well. So that's a whirlwind tour of the winding road to translation. I left about 20 minutes. Happy to answer any questions you might have. Yes? For normal tissue injury? Well, what we did see is we saw a lot of local inflammation. So around the site where we injected, it got red, it got tender. It tended to last a couple days. A lot of the patients developed flu-like symptoms, fever, chills, and some of them got pretty sick. Some of them had to get a couple liters of fluid to get through that. Within a few days, they seemed to be okay. We did not see any evidence of end organ damage from the immune system yet. I suspect we will, but we haven't in the small number of patients we've looked at. Yes?
Pretty similar to the complications, but I'm wondering if you have suffered any complications of intra-tumor injections,
Yep.
like metastasis?
Well,